everybody, we're going to start talking about a physiological topic in the nervous system, which is called the nerve impulse transmission. So in order to understand nerve impulse transmission, we are first going to discuss resting membrane potential. In your course pack, this information is found on page 95. In order to understand resting membrane potential, let's talk for just a moment about typical neuron structure. You're looking at a multipolar neuron in which you see dendrites branching off of the cell body. We see a cell body here that contains the nucleus and organelles. The axon hillock right here connects the axon to the cell body, and then the axon here is unmyelinated. You'll notice the impulse direction is from the dendrites toward the cell body, from the cell body down the length of the axon to those axon terminals. The neighboring cell that we're going to transmit a nerve impulse to is called a postsynaptic cell. The axon and the neuron to which it belongs is called the presynaptic cell. So what we're going to discuss is how we conduct the nerve impulse down the length of an axon. We're going to assume that it's an unmyelinated axon, and then we'll also talk about transmission at the point of synapse. In order to understand all of these steps, we first need to talk about resting membrane potential. In other words, what does it take for a cell to stay at rest? Now, interesting point here. If you think about making new connections through perhaps studying anatomy and physiology content, you have to give time for these axons and dendrites to grow together. If you cram the night before, you don't give time for these axons and dendrites to make their connections. In other words, you might have an axon sitting here and the dendrite of a neighboring cell too far away to make a connection. Over time, these two will grow closer together in order to make a synapse between them. So when you study the night before, no wonder you can't remember anything, right? Because you're not giving time for these synapses to grow and make new connections. Studying repeatedly and all along gives you time to make those connections. So this is going to be part of our conversation, is what happens at a synapse between an axon terminal and neighboring dendrites. So let's talk about resting membrane potential. In other words, resting membrane potential is what it takes for a cell to stay at rest. Our conversation is going to focus on neurons. So first, let's talk about the types of ions that are involved here. We are looking at a polarized neuron in which we have mostly sodium ions. Of course, they're Na positive, right? They're cations. Outside the cell, okay? So extracellular fluid means that we are looking at the outside of the cell. If you count, you can see that there are more sodiums outside the cell than there are potassiums. Potassiums are, of course, K with a positive symbol there. That means that those are also cations. Now, the cations are both scattered on either side of this membrane. Potassium's mostly inside, sodium's mostly on the outside. Now, when we talk about a polarized neuron, we talk about one that has an unequal distribution of ions on either side of that plasma membrane. So we actually have more ions on the outside. That's why we have a positive charge here on the outer surface. We actually have less ions on the inside, and so that's why we have an overall negative charge here inside the cell. So it's not that we have cations and anions that we're dealing with. We're actually dealing with cations, positively charged ions, in both locations, both outside the cell and inside the cell. So it's not that we're dealing with positives and negatives, it's that we're dealing with the outer surface of the cell, which is more positive because there are more ions compared to the inside of the cell, which has, of course, still positively charged cations. There's just less of them. So it gets really confusing saying more and less all the time. So we make this a very simple black and white issue, and we say outside the cell has more ions. Wherever there's more, you have a positive charge. Wherever you have less ions, you have a negative charge. So at rest, you have positive charged potassiums inside, positively charged sodiums outside. But the positive exterior environment is because there's simply more sodiums on the outside surface. Okay, so you may want to use this little device over here on the left side of your screen, SOPN which helps you remember that at rest, sodium is outside the cell. That carries a positive charge outside. Potassium is inside, creating a negative charge. And again, let me repeat, it is about where you have the majority. The majority of the ions are outside, and that's why we have an overall positive charge there. So let's go back to this term, polarized. Polarized means that we have an unequal distribution of charges. We actually have more ions in general outside the cell than we do inside. So at rest, we say a neuron is polarized. So here's a sneaky little story. Those potassiums and sodiums don't like to stay where they are. They like to travel across the membrane when they can and either enter the cell or exit the cell. So they're constantly sneaking across the membrane if it's possible for them to do so. In order to maintain our resting potential, we need a little bit of help cleaning the place up constantly. So here it comes into play a sodium-potassium pump. It is in fact an integral protein. You'll notice it sits across the entirety of the plasma membrane, and it's powered here by ATP. So it's constantly working to remove sodiums from the cell, to move them back outside the cell, and to replace the potassiums back inside the cell. So that's of course where we expect to find them at rest. We expect to see sodium outside the cell and potassium inside the cell. Now here's the catch. Notice how many we're moving. We are moving 3 sodiums to the exterior of the cell. How many potassiums are we moving back in? Only 2. So why not move an even number? Why not move 3 out and 3 back in? It all has to do with maintaining a polarized environment. In order to keep the outside of your cell positively charged, you have to have the majority of your ions out there. So that's why we move out 3 positively charged sodiums and we move in 2 positively charged potassiums. So that way we maintain a polarized state where we have sodiums outside the cell and the potassiums inside, but we maintain that unequal distribution of these charges. Now, I think of the sodium-potassium pump kind of like a sump pump in a basement. If you've ever heard of a sump pump, it keeps the basement from filling up with water, and it's always on. This sodium-potassium pump works at a particular speed, only so fast, and in order to maintain resting potential, it's constantly moving those sodiums that trickle in or potassiums that escape out back to their original location to maintain a polarized state. Keep in mind, polarized means we are at rest. This cell is not conducting a nerve impulse when it's at rest. Let's talk about the negative charge of a cell when it's at rest. When a cell is at rest, we say it has a negative 70 millivolt charge. That is a good number to know. Okay, so at rest, you'll notice that we are looking at a negative 70 millivolt charge. That tells me how many potassiums are inside versus how many sodiums are on the outside of that cell. So it's the distribution here of charges that maintains a negative 70 millivolt charge at rest. Okay, so we say that's an electrochemical gradient in which potassiums ideally would like to move along their concentration gradient, as well as sodiums would ideally like to move along theirs. Here again, you get a look at this sodium-potassium pump right here. Sometimes it's called the Na plus K plus pump. It is ATP powered. That means it requires energy to do work. And how many sodiums are moved out? 3, right? For how many potassiums moved in? Just 2. So this uneven distribution of charges is how we maintain a resting potential here. Notice it's more likely for the potassiums to escape. You can see here potassiums may diffuse out. Some sodiums may in fact come in, but for the most part the membrane is somewhat impermeable to their movement. We really rely on channels to allow those to move greatly from one place to another. All right, so here's another look at the plasma membrane. As I was mentioning, there are sodium channels that allow us to open up and let sodiums flow. And there are also potassium channels here. As long as those channels are closed, for the most part, sodiums and potassiums can't go to their areas of low concentration. So notice here at resting potential, or the resting state, all gated sodium and potassium channels are currently closed. That means that we're not looking for them to migrate from areas of high concentration to low concentration. Notice at rest, where sodium is mostly housed, it's outside the cell, right? And of course, at rest, potassium here is inside the cell. So by keeping these channels closed, it makes it a little harder for them to migrate from areas of high concentration to low concentration. Now, since these are channels and not pumps, they simply need to open up, and they operate passively, meaning no ATP is required. That means that we can allow for these ions to move from areas of high to low concentration once those channels are open. So unlike the pump, these channels here can simply operate by diffusion. All right, so what is the charge at rest? It is negative 70 millivolts. That's a good number to know. And of course, if we're at rest, we're looking at sodium and potassium channels staying closed. But what is this pump constantly doing for us? It maintains resting potential. How does it do that? It moves out how many sodiums? And it moves back in how many potassiums? Okay, so the reason we're moving an uneven number, 3 versus 2, is all about maintaining that resting state. That gives us an overall negative 70 millivolt charge for a neuron or a cell here at rest. So I just want to take home some messages for you. First of all, a cell at rest is a polarized cell. That means there's an unequal distribution of ions on either side of the membrane. That's how we create an overall negative charge inside and an overall positive charge outside. To keep this simple, the conversation goes like this. The cell has an overall negative charge inside due to less ions present there in general. And outside the cell has an overall positive charge simply due to more ions. There's other things at play here. There's anions, there's proteins, but for our conversation, we're going to keep it simple and think about it in this way. Sodium ions are mostly outside the cell. That's where they're more highly concentrated at rest, while potassium ions are mostly inside the cell. Again, that's where they're mostly highly concentrated at rest. They can move through open channels, but normally during resting potential, those channels should be closed. So in order to maintain order, the sodium-potassium pump returns stragglers to their locations. Any sodiums that come in are pumped back outside. Any potassiums that escape are returned back inside. Keep in mind, it's 3 sodiums for 2 potassiums. By making it an uneven number, we maintain a resting potential of negative 70 millivolts.